Okay, thanks for the invitation. My talk today is about the neural relation inference to learn long-range allosteric interactions from molecular dynamic simulations. Yeah, many proteins so functions are regulated by dynamic processes just like protein folding, protein unfolding, protein activation, or allosteric. So the allosteric ligand binding affects the active site remotely by, by spatial long-range intra protein communications. And the molecular dynamics simulations, MDs, is a powerful computational tool to handle the allosteric effects by qualitatively and quantitatively analyze the conformational changes related to its function. And it simulates the physical movements of the atoms and the molecules by Newton's equation of motion. But it may fail to capture the reasonable functional information due to the limitation of the time scale of simulation. The trajectory obtained from the molecular dynamic simulations are usually high dimensional. So it contains the movements of many atoms at many time points. It is a kind of a challenge to interpret what behind the trajectory. And there are two classical methods. One of them is PCA, principal component analysis, and another is the famous cross correlation analysis. Usually, these methods work well in many cases, but they assume the system is near equilibrium and they are limited by their only capture the linear relations. Okay. However, the molecular dynamic driving forces, such as the van der Waals energy, bound angle energy, and dihedral angle energy, are not always linear. And the dihedral angle energy involves atoms other than pairwise interaction. And in the meantime, these driving forces may not be accurate due to the limitation of their measurement. So here we introduce the neural relation inference NIR from the computer vision to address this challenge. So actually the NII is an unsupervised model to infer interactions while simultaneously learning dynamics purely from the observational data. It can find the interpretable structure and predict complex dynamics. So one toy example is on the left. This is a dynamic system of 5 moving particles. So some of the particles, they have relations with each other, while some are not. And our goal is to try to identify which particle has a relation with which particle. Observing the trajectory of these particles, the underlying interactions may be modeled as an interaction graph. Say the trajectory of the red is related with the trajectory with the orange. Then we add an edge between the red and orange. And the trajectory of the orange does not have the relationship with the trajectory of the purple. So there's no edge between the orange and the purple. And one successful application of the NIR in computer vision is analyzing the movies. So with the training of NBA players playing basketball, it successfully identifies some players' preference using either right hand or the left hand. So we can see, so the hand has been highlighted with more edges than other point of the human body. Based on that, we propose the NIR-MD model, neural relation inference molecular dynamics simulation. Of course, we need the molecular dynamics first. And the model uses the trajectory of the residue positions as the input. And it models the dynamic systems in different time steps. It uses an encoder to learn a distribution and reconstruct the trajectory through the decoder. The learned distribution can be used to infer the edges, and then we have the interaction graph. Here are some details of the model. The input is the trajectory of the residues, and in each time point, the coordinates of x, y, z in the 3D spaces, and the velocity in x, y, z are treated as the input feature. And in the beginning, we need to build a full graph as a skeleton between each residue pair. And then we use a encoder as the message passing graph neural network, passing information from node to edge and edge to the node. And z given x is the target concrete distribution. That is our goal. And after sampling, if the probability of Zij is more than 0.5, and then we have an edge between I and J. And the decoder is also a message passing graph neural network organized in recurrent neural network fashion to reconstruct all the trajectories. So the training goal is to minimize the differences between the input trajectory and the predicted trajectory by the model. In the case study, we use the APO Pin1 with molecular dynamics simulations to study the inter-domain allosteric regulation. APO Pin1 is a typical therapeutic target which regulates the cell proliferation, responds to immune, and often treated as a drug target. And we know some mutations like the I28A, it will weaken the inter-domain interaction and reduce the catalytic activity. In the study, we systematically explore 3 scenarios in the system, and we run molecular dynamic simulation on each of the scenarios. So here is the first scenario in red. So in the beginning, the WW domain and the PPI domain, they do not interact with each other. And we can see the red lines are the true trajectory from the molecular dynamic simulations. And the blue are predicted trajectory reconstructed by the ISMB model. In the second scenario, with the binding, these 2 domains interact with each other. And in the third scenario, with the point mutation as leucine mutated to aniline at the 28th residue, these 2 domains interact. So that will break the interaction between the WW domain and the PPI domain. We can see in all these 3 scenarios, the NIR-MD model reconstructs the trajectory well, and that means it may capture some relationship behind the trajectory between the residues. And here is our output, the learned edges from the proposed model. So the outlines of the heat map are the learned edges in the residue level. So both x-axis and the y-axis, they are residues. So if there's a value there between the residue at x and the residue at y, then there is an edge between these 2 residues. And in the bottom, are the learned edges in the domain level, abstracting results in the protein domain. So we can see huge differences in this learned interaction graph. So this is the first scenario, the WW domain and the catalytic loop domain, which is the most important domain in the protein. They do not have any edges there. With binding, they have edges. And the edge disappeared when we have the mutation. So actually, this phenomenon is consistent with our prior biological knowledge, which is fantastic. And here is the interaction graph with directions. So this is the direction from the sender to receiver. And this is the opposite. And with the inferred interaction graph, we can infer the inter-domain allosteric communication pathways by calculating the shortest distances using the most famous Dijkstra's algorithm in computer science. So we can see with the binding, we have a whole pathway from the start point as the WW domain to the end point at the catalytic loop. And we can see the C. So if there's a mutation at the residue 28, the whole pathway gets interrupted just at that position. One feature of the proposed model is it can capture the signal sooner. Comparing with the Hessian-based derivative centrality metric, which captures the signal at the frame 3000, so which is 300 nanoseconds over the molecular dynamics simulation. The proposed NIRMD captures the signal as early as 500 frame. Yeah, so that is only 50 nanoseconds over the molecular dynamics simulation. Another feature of the proposed model is its free energy estimation. So the constraint network analysis, CNA, is another popular tool to build the neighbor stability maps reflects the local stability of the residue context. They provide a simple criteria to estimate the free energy. And I think we can do the same thing with the interaction graph we inferred. Of course, they have a different meaning. So the local stability from CNA and the interaction inferred by our NRI-MD model. Here is an analysis scan study on PIM1 system. So in that, people do mutation from Y type to aniline on each residue, and it measures the thermodynamic measure delta G to quantify whether the mutation will increase the protein stability or will decrease the protein stability. We can see each mutation NIR-MD inferred different interaction graph. And I think if we go to detail, just like the mutation at the 7th residue is totally different from the interaction graph we get from the 9th residue. So one is stabilizes the protein, and the other is destabilizes the protein. And we can see our method get a very good results in prediction of free energy change. We can see most of the data sites are around the diagonal with the Pearson correlation 0.94. which is very good. And it is better than the compared method in this case. In summary, so the NIRMD model is one of the first attempts to use a graph neural network to analyze molecular dynamics simulations. And it effectively generates the interaction graphs through an embedding over reconstructing molecular dynamic trajectories. And it accurately estimates the free energy changes. And it's infer the pathways mediating allosteric communications. And the code is publicly available at GitHub. And I want to thank my supervisor, Professor Dong Xu at University of Missouri, and my collaborator, Professor Wei Han at the University of China, and the co-first author, Juexin Wang, who did most of the molecular dynamic simulation works. And the paper was published months ago. And as an incoming assistant professor, I'm recruiting PhD students who is interested in deep learning and graph modeling, molecular biology, and single cell biology. If you are interested, come to me. And that's my talk. Thanks for your attention, and I'm happy to take questions.
First of all, really great talk. Thank you so much. It was a great method. So your interaction graphs are at the level of domains, right? So if I am not mistaken at the latent variable model, I mean, after encoder, you'll have individual domains as a, so does that require you to annotate and know all of the domains of a protein? And what about the residues that are outside, annotated domains? Do you consider that too in your interaction graph?
Yeah, thanks for your question. So actually, if I can go back, so let's go back to here. So actually here, so we can see we inferred several edges between different residues. So the outline is kind of detailed things. So just like the red rectangle, this is something we can interpret, but we can see lots of other things that we cannot interpret. Yeah, so probably we only probably here in these three scenarios, the bottom things is what we can interpret now, and for the other things, that's probably ways to go. And another thing about the domain, yeah. So for that part, I think that is just for the abstraction of the information. So if we plot everything in the residue, that may be too messy. And if we come back and we know something about that pin1 system, and some residues are in the domain, and so that will help. That will definitely help us to build the knowledge there. So with the things bottom line.
I see. Thank you so much for the clarification.
Thanks.